everyone. Welcome back to my channel. This is Guru Marunong, Kinig, Tutok, at Mating Marunong. Our topic for today is all about DNA replication, transcription, and translation. So let's proceed to our topic. How protein is made using information from DNA. Protein. So protein is the most varied molecules in which the human body contains at least 1,000 different kinds of protein. When we say varied, pinakamarami na. Siya, pinakamarami. Varied. Marami siyang variety. Iba. Then, protein is made up of amino acid. Therefore, amino acid is the building blocks of proteins, which are made up of long chain of chemical units. Importance of proteins in the body. So protein hormones regulate many physiological processes, like insulin, for example, that affects glucose transport into the cell. Protein in the blood help us blood clotting factor and transport molecule. For instance, another example, hemoglobin transport oxygen in the blood. Protein act as ion channel, carrier, and receptor molecules in the cell membrane. Protein synthesis. When I say protein synthesis, is making proteins. So there are three kinds of RNA in protein synthesis. First, let's have the mRNA, or the messenger RNA. So messenger RNA is the type of molecule RNA that travels from nucleus to ribosomes. So alam niyo na kung saan gaganap ang protein synthesis sa ribosomes, in the cytoplasm, where the information in the copy is used for protein product. So itong mRNA, later on our topic, makikita nyo siya kung paano niya may transcribe yung message or yung sequence from DNA to RNA. Ribosomal RNA, or rRNA, is the RNA component of the ribosomes and cells protein factory. So pag sinabi natin RNA, siya yung RNA component sa ribosome. Siya yung protein factory. So ang ribosome cell protein factory natin in all living cells. It provides a mechanism for encoding mRNA into amino acid and interacts with the RNA. So siya yung nag-help kung paano magiging amino acid ang mRNA through the use of tRNA, which is the transfer RNA, or tRNA. Is an adapter molecule composed of RNA, typically 73 to 93 nucleotides in length, that brings amino acid from the cytoplasm to the ribosome to help make growing protein. So yung tRNA naman, or transfer, siya yung nagdadala ng amino acid sa cytoplasm to ribosome. So from cytoplasm to ribosome, yung tRNA ang nagdadala ng amino acid. So later in our topic, discuss din natin kung paano nadadala ng tRNA from the cytoplasm to a ribosome to help growing protein. Next, DNA versus RNA. So DNA is double helix. Pag sinabi natin double helix, para siyang ladder. Ganyan ang structure ng DNA. Then RNA is single helix. So kalahati lang siya ng DNA. DNA uses the process of replication. So later on our topic, we will discuss about replication, transcription, and translation. So pag DNA, dito lang siya naka-focus sa replication. Pag RNA naman, siya yung essential o kailangan para sa transcription and translation. DNA uses thymine, adenine, guanine, and cytosine, while RNA uses uracil. So ito lang yung pinagkaiba ng DNA sa RNA. Yung thymine, papalitan siya ng uracil. Next, DNA replication. It's a biological process of producing two identical replicas of DNA from one original molecule. So from the word replicate or replica, magpuproduce siya ng two identical replicas o two copies ng DNA from one original. Galing lang sa isang DNA molecule. So paano mangyayari yun? So dito na yan sa DNA replication. So DNA replication takes place in the cytoplasm of prokaryotes. So pag prokaryotes, cytoplasm. Pag eukaryotes, sa nucleus. The enzymes start to unzip the double helix as the nucleotide base pair separates. So, ito yung dalawang base pair. This is the base pair. Ito yan. Dalawang yan. Base pair. So, kung nakita nyo, it start to unzip. So, naghihiwalay sila. Each side, double helix runs in the opposite direction on both strands of the molecule. So, naghiwalay sila. Or nag-unzip parang sa zipper, naghiwalay. Nag-unzip. Next, DNA replication. Free nucleotides pair with the base exposed as the template strand continues to unzip. The enzyme complex DNA polymerase attaches the nucleotides together to form a new strand of similar to each template. So, nag-unzip siya, then yung polymerase attaches. So, didikit na silang dalawa. Ito yung tinatawag na so, ito na yung magiging new strand mo ng DNA. So, as you can see, nag-replicate na siya or napabuo na siya ng dalawang bagong DNA copies. So, notice here, nakikita nyo, yung thymine is paired with adenine. So, ito yung original base pair. So, lagi kayong titingin sa original base pair. Siya yung original base pair. So, dito rin, dito naman sa kabila, yung adenine, pinair naman siya sa thymine. Dito naman sa kabila, yung guanine, pinair siya sa cytosine. Dito naman yung cytosine, pinair sa guanine. So, vice versa lang sila. Tulad ito, adenine pair with thymine. Thymine pair with adenine. So, nakapagbuo na siya ng 2 copies of DNA strand. So, itong DNA strand na to, subunit of molecule of DNA polymers profits the new DNA and the DNA ligase enzyme seals up the fragment into one long strand. So, makakabuo na siya ng panibagong or nag-replicate na yung DNA natin. So, sa pamamagitan ng ligase or yung enzymes na ligase, isi-seal up niya yung DNA or pagdidikitin niya. Then after that, two similar strand molecule of DNA result from replication. So yung nagpakadalawa na tayong copies ng DNA, the new copies automatically wind up again. So yung ito na yung new copies natin. So nakabuo tayo ng new copy. New copy. So yung new copy na yan, maghihiwalay ulit siya. Then, magpe-pair ulit siya ng panibagong DNA strand or base pair. So, hanggang dumami siya ng dumami. So, that's how DNA replication works. So, paulit-ulit lang siya. Tapos itong uling new copy na to, maghihiwalay ulit siya. Then, papartner ulit siya ng panibagong base pair hanggang dumami sila ng dumami. Next, DNA replication continued. So, thymine is paired with adenine. So, katulad yan, sabi ko kanina. And cytosine is paired with guanine, vice versa. So, vice versa sila. So, yung thymine, lagi siyang partner with adenine. And adenine, partner siya sa thymine. Ganon din sa cytosine to guanine, and guanine to trypsin. So, yan. Makita nyo. Thymine, adenine. Adenine, thymine. Dito naman sa baba. Adenine, thymine, thymine, adenine. And guanine to cytosine, cytosine to guanine. Next, transcription. So, the DNA is found inside the nucleus of the cell, which are embedded into the chromosomes. The genetic information within the DNA must be transported to the ribosome in the cytoplasm where protein synthesis takes place. So nakita natin dito, ribosomes where protein synthesis take place. So sa ribosomes, take place ang protein synthesis. The genetic information or code is copied to mRNA through the process of transcription. So dito na natin malalaman kung paano matatranscript ng mRNA yung DNA. The transcription process occurs when the nucleotide sequence along the DNA is copied into the mRNA. So, ganito siya mangyayari ngayon. So, ito yung DNA strand natin. Iwalay sila. Then, papartneran siya ng mRNA. So, this is the mRNA. So, notice here, kikita natin yung thymine, partner pa rin siya sa adenine. But, the adenine is partner to the uracil. So, magkaiba siya pag sa mRNA. Pero yung guanine to cytosine and cytosine to guanine pa rin. Is another example, adenine to uracil, adenine to uracil. Pero pag thymine, sa adenine pa rin siya makapartner. Next, translation. Translation is the final step in the synthesis of a small protein through the help of mRNA. The transfer of code from mRNA to a small protein begins when the mRNA molecule attached to the ribosomes, which form the mRNA ribosome complex. The different amino acids found in the cytoplasm must first be transferred in the ribosomes complex. An amino acid is then attached to a specific tRNA. There are plenty of tRNA as to presence of amino acids because it is intended that each tRNA is coded to a specific kind of amino acid. So, ganito ang mangyayari sa kanya during translation. So, itong tatlo na to, example, ito, hahanapan siya ng tRNA. Then yung tRNA, magiging amino acid. So, here's the tRNA. tRNA yan. So, yung tRNA na yan, may kasama na siyang amino acid. Then, a-attach siya sa mRNA. Then, yung mRNA, then yung tRNA, lalabas ulit siya. So, hiwalay na siya ito, magiging amino acid na. So, ganun din itong tatlo na to. Papartner na ulit siya ng tRNA na may kasamang amino acid, katulad nito. Then, aalis ulit yung amino acid. Then, yung mga amino acid na yan, magpo-form yan ng long chain. Then, makakaproduce na siya ng protein. So, translation is the converting of information from RNA or mRNA, itong mRNA na to, into protein. Test your skills. So, pwede nyo i-post itong video kung gusto nyo magsagot. So, protein synthesis takes place in the process of converting RNA into protein is called what enzyme seals of DNA fragments into one long strand? What is the process of producing two identical replicas of DNA from one original DNA molecule? What is the final step of synthesis of a small protein through the help of mRNA? So, I give you a minute para i-post itong video na to. Then, check nyo kung tama yung sagot nyo. So, yung number 1 natin is ribosome. Number 2, translation. Number 3, ligase. Number 4, DNA replication. And number 5, translation. Tama ba ang sagot nyo? Next, replicate the DNA sequence. So again, I will give you a minute para i-post yung sagot itong video na to para sagutan tong DNA sequence ito. So here's the answer. Next, transcript the DNA sequence. So ito, transcript nyo naman siya. At ito ang sagot niya. So, maraming salamat sa pakikinig. Naway natuto kayo. Maraming salamat sa inyong lahat. Goodbye.